Good morning. I'm Dirk van HYFT. I'm the CTO and co-founder of BioStrand, a subsidiary of IPA immunoprecise antibodies, and that's an antibody development company. So we have both the in silico part and the wet lab capabilities to develop antibodies. And so I will present today the shift from wet labs to GPUs. So, and to explain how we can accelerate life science through further computational transformation. And of course, there's quite a lot of things going on about AI. And the big question is AI pushing biology, or is biology applying AI? And so we really advocate about building a bio-native AI framework. So really based on the biology, on the complexity of biology, building a world model that describes all the complexities that you have in biology. And on top of that framework, apply AI so that we can really capture more meaningful insights. And as I said already, we are in the business of drug discovery. And that means that once you start with a certain drug target, of course, it's a multi-step process. It's a very complex process. And as you say, there are many ways to go to Rome, but there's also many ways to go to Paris. And so there are quite a lot of solutions. And that is the key thing. When you try to find the right solution, you have to gather quite a lot of very specific information. And so what is really a challenge is to balance between molecules that have a very good functionality, but also reducing the clinical risk so that you also don't harm your patients. And that typical process of developing drugs takes quite a lot of time. In the current days, it can take maybe even 10 years before a certain component gets into the market. And so it's a really long and extensive and also expensive trajectory. If we look at the day's, let's say, market and the players that are active in drug discovery, then you really can see that it's all about wet lab. So the big pharma companies have very big wet lab capabilities. And of course, there are also some drawbacks with this wet lab capabilities. First of all, it's quite human intensive, but also capital intensive. And there is a limited amount of components that you can test. So the throughput in such wet lab capabilities is relatively limited. And also, it takes quite a lot of runtime and time to find the right solution. And that's why we have developed our platform to facilitate this and to enhance that process, so that you can deliver and find drug targets in a much shorter time and also test more components. If you look at the drug target environment, then you can divide it in two big groups. Well, you have what we all know, the small world, the small molecules, so that are the classical drugs, and therefore, well, you have relatively straightforward, let's say, AI tools. It's also the data is relatively easy to get. And they also have quite standardized descriptions. While in the area that we are playing, it's really in the biologics. So it's really the protein world. That's much more complex. And you need to take much more features into account to develop a certain drug target. And that's why we have our HYFT-based technology. So it's really an indexing system. And with that indexing system, we can combine sequential data, like genomic data, structural data from the 3D proteins, for instance, but also functional data that come from wet lab experiments, but also what's written in scientific literature. And also what is very important to understand that this indexing system can accept or add information from different large language models and integrate that information. So you have an indexing system that can look again at sequence data, structured data, functional data. You can aggregate information from different large language models and also with contrastive learning to see to update this model. So that means you have really a world model of biology. It's not just statistical inference, it's a huge network. And that gives us much more insight in the biology. And for instance, one of the examples that we are also very proud of is, for instance, that we can model what they call epitope mapping. And what is epitope mapping? For instance, if you have a certain target for a certain cancer, and then you have antibodies, you want to know where the target and the antibody is binding. And here we have proven that indeed we can have near X-ray precision with our methodology. And also if you want to do, for instance, X-ray, that costs a lot of money, but also takes quite a lot of time. And so we can shorten that process, and again, with the same precision, or almost the same precision. So that's why we have built our platform. Our platform, it's a SaaS platform, where we have different AI tools, AlphaFold, Vultr, immunogenicity screening, affinity maturation. And what is also important to understand that we integrate all that information that's coming from these different components. And based on that, we built a huge knowledge graph, and that helps us to understand, let's say, the biology. And in that way, we can screen lots of data, all candidates. And there are only a few of these candidates that can go to the wet lab. And in a certain way, that's also what you see, the transformation in this field. In the past, you had these big pharma companies with these huge wet lab capabilities, and now there are more the AI companies that start building drugs and use lab capabilities. So it's really a transformation. It's a really shift in this field. And so again, what is key of our technology, what sets her apart, is that we have discovered, and it was really discovered, HYFT patterns, so that are in a certain way the words in biology. And with these HYFT patterns, we have the right tokens to apply to these large language models and integrate all that information. And so with these 660 million HYFT patterns, we can index whatever exists in the biosphere, going from plants, viruses, certain cancer targets, and so on. So again, it's a very general approach to index all the information that is in biology and associate on top of that information from different large language models. So it's a multimodal system. And so we are also very proud that we have developed our bio-native AI framework on a Vultr platform. And it's really worthwhile to look at some of the blocks and the benchmarks that we have done together with Vultr on different areas. So we have compared EMD versus NVIDIA in text analytics, so how to create embeddings from text. We did it also in protein, how we can create protein embeddings also in comparison with using EMD and NVIDIA accelerations. And also what is a very new area is what they call RFdiffusion, so really in the de novo. And so the starting building really proteins from scratch. And again, very nice results that we have, and I really refer also to the different blogs that we have written around that. And so together with Vultr, we are really building and bridging biology and AI through compute. And that's the way to move forward. That's the way to move forward in this field and to make safe drugs for patients, but also very precise drugs for patients. Thank you very much.